Hi, welcome to the Neuro Revision Lab. Today we're going to take a look at microscopy. That is, the use of microscopes and the calculations that go along with it. The things you're first going to need to know about are plant and animal cells, and you're also going to want to be able to do simple calculations. Remember, in GCSE, they are always going to let you use a calculator in a science exam. You're going to be able to describe how microscopes have improved and explain why electron microscopes show tiny details inside cells. And you're going to be able to calculate sizes using the magnification formula and use the correct units to go along with it. The key words you're going to need to use are light microscope, electron microscope, magnification, image size, actual size, micrometer, and nanometer. Don't forget to make a special note of the units being used here. Take a moment also to note those down in your books. Practice sounding them out, listen carefully to how the words are used, and research their definitions later. Microscopes have been around for hundreds of years. What started off as an interesting scientific experiment ended up becoming vital in medicine, environmental science, microbiology, geology, and a dozen other applications across all of science. Now they are so powerful that you can see in unbelievable detail inside cells. And electron microscopes go even further, though they do have their limitations. Each improvement has brought stronger magnification and higher resolutions until we finally reached the electron microscope. The key difference between a light microscope and an electron microscope is that they don't use light. Instead, they work by firing electrons at the subject. Unfortunately, living things don't survive the process, but the magnification and resolutions achieved are incredible. Label this diagram of a microscope into your revision notebooks, and also make sure you record this equation. I find the best way to remember it is as if you're aiming your microscope at cells. Actual size, image size, magnification. They spell out AIM, as in aiming. Your notes at this point should probably look something like this. Take a moment to make any corrections you feel like you might need before we move on to an actual exam question. Remember, this is just a suggestion of how your notes should look. They are just that, your notes. Okay, so let's attempt a really good common exam question. Figure 1 shows an example of a cheek cell. You can see that a measurement of 50 millimeters has been made. As this is inside of the microscope, it must be the image size. So let's start that question. Question A. The actual size of the red blood cell is 10 micrometers. On the diagram, the image size is measured as 50 millimeters. Calculate the magnification of the image. Use the formula. Magnification equals image size divided by actual size. Question B. State one advantage of using a microscope to study cells. Okay, and a little bit of a nudge to help you get started. Highlight your command words. What does the question actually want you to do? Then highlight your keywords. These are the same ones you would have noted down earlier. And finally, your values. These will be the numbers that you're using for your calculation. Feel free to pause the video and spend a little bit of time on this. The answers will follow shortly. For more revision resources, tips, and helpful materials, please visit the NeuroLab store. Everything here is specifically selected or made for neurodivergent learners. And as an added bonus, each purchase supports the channel, allows us to keep producing video content, and as usual, subscribe, like, and share. And here we are. Let's check out these answers. you can correct and try again if necessary. Question A asked us to calculate the magnification. So the actual size of the red blood cell is 10 micrometers on the diagram. The image size is measured as 50 micrometers. So we're going to have to convert our 50 millimeters actual size into micrometers first. And then all we have to do is remembering our AIM equation as actual size divided by image size, and that will get you 5,000 magnification. Question B asked us to state the advantage of using a microscope to study cells. And hopefully you said something along the lines of, it enables us to be able to see small structures or cells that would otherwise be impossible to see with the naked eye. This is the kind of question that often comes with a little bit of interpretation and multiple answers for the examiner. If you're finding the channel helpful, you can pick up one of our specialist neurodivergent notebooks designed to make learning clearer and easier. They make perfect additions to EHC plans and are not only comfortable to write on with off-white pages, but are also broken up according to the Cornell note-taking style. They can be used in schools and at home as dedicated revision notebooks. Like and subscribe to the channel for more SEN-focused science revision. Click on one of the links for your next video, and we'll see you next time.